Hi, everyone. Thanks so much for joining us today. Welcome to today's webinar, Why SMLM for LNP Research with ONI. My name is Carolyn Hartman, and I am going to be the host for today's webinar. But just before the webinar begins, we just want to go over a few house rules for the webinar. So I'm sure that you've all noticed by now, but you do have a webinar navigation panel on the side of your screen. So this is containing a few different sections. If you'd like to clearly see a specific section a bit more, you can click on the expand icon. It's located in the top right corner of each of the sections. Today's webinar will last about 50 minutes, leaving approximately 10 minutes for Q&A at the end. And you are welcome to submit any questions that you have throughout the webinar via the chat section, which is located within your panel. However, Stefan will only be answering your questions during the Q&A session. If he is unable to get to your questions, then we will forward him both your question and the email address in which you registered with. So within your navigation panel, you will also find a handouts section. So in here, you can find our company brochure, and you can also find the LNP brochure, just touching base on some of the topics you're going to learn about today. So please feel free to have a look there or on our website located at www.oni.bio. And please note that today's webinar is being recorded. So within the next few days, you are going to receive a thank you for attending email with a copy of today's webinar to refer back to. In addition, a video recording of today's webinar will also be stored on the resources section of our website. And now I would like to introduce you to our wonderful presenter for the day, Dr. Stefan Balint. So Stefan is currently a senior scientist at ONI, and after earning his PhD in biophysics, Stefan moved to pursue his career in SMLM, where he focused on the development of the correlative image method based on single particle tracking and super resolution microscopy. And before joining ONI, Stefan used his knowledge in super resolution microscopy to answer open questions in immunology. And by using multicolored D-storm imaging, he discovered that supramolecular attack particles, so a tactile arsenal of T cells secreted to the immune synapse. And with that, I'm just going to pass the presentation over to you, Stefan, to begin the webinar.
Thank you, Caroline, and welcome everybody to the webinar. So today, as Caroline already mentioned, I will give you some insight how actually single molecule localization microscopy can help your LNP research. So today's webinar will consist on the following topics. So in the beginning, I will give you a small brief introduction about LNPs. So what are the LNPs? What is their composition? And then I will also follow up with some roles and their importance in research and medicines, where I will highlight some key questions that are still remaining and needed to understand before moving to clinical trials. And with this small introduction to lipid nanoparticles, I will introduce you to single molecule localization microscopy, and then I will guide you through different workflows and how actually the single molecule localization microscopy can help you in your lipid nanoparticle research, and how can you advance your basic research before you move to clinical trials. And at the end, I will also give you some insight about the data analysis that we as ONI provide with our nanometer system. And at the end, if you'll have some time, as Caroline mentioned, for the last 10 minutes, we'll have some questions to Q&A. But before we start, let's take a small poll.
Just wait a few more seconds to get a few more answers in.
Okay, I can't see the poll. I'm not sure.
All right, so it looks like 77% said no experience, 23% said some experience, and no one identified themselves as an advanced experience.
Great. So I will give you an insight to it, how actually single molecular localization microscopy can very simply help you to characterize and analyze the lipid nanoparticles. So as I mentioned earlier, let's give a little bit of small introduction. What are lipid nanoparticles and how they actually are characterized? Because sometimes these particles actually can be mixed with liposomes. However, there's a huge difference between these two particles. So lipid nanoparticles are known to have a monolayer, a lipid monolayer. This is a major difference between the liposomes and lipid nanoparticles. As liposomes are formed by a double layer, so there's a bilayer instead of monolayer. But then the lipid nanoparticles, or LNPs in the short term, are actually can be varied in terms of lipid composition. So the major components of these lipid nanoparticles are polyethylene glycol and also cholesterol. However, the composition of this LNP is actually customized. And different companies who are actually using these lipid nanoparticles are actually having their own formulation, which can be easily tuned by modifying the amount of polyethylene glycol, for example, or changing a different lipids within nanoparticles. So all these lipid nanoparticles are actually also containing a core. And the core of these lipid nanoparticles is used as a cargo system. So the lipid nanoparticles in general are actually delivery systems which can hold some proteins, biomolecules, or even nucleic acids inside their core. Mainly, as you guys already know from vaccines, these lipid nanoparticles are actually often containing RNA or mRNA within their core. And as I already mentioned, there's a lot of customization happening in these lipid nanoparticles. As an advantage of using these lipid nanoparticles, as they are pretty easy to prepare, but then they also need to be customized. So a lot of these lipid nanoparticles are used for cancer research currently, but then they need to have some targets. So to be very specific to your target of interest, you are actually you can modify the surface of these lipid nanoparticles, which become ligand specific. I will come to it a little bit more deeper later on. So this is just a brief introduction of lipid nanoparticles, but what is the role of these actually particles in research and medicine? Actually, how much we actually know about that. So lipid nanoparticles are starting to be widely used in research. And if you look for a number of publications by searching all lipid nanoparticles or LNP, you can see that there is an increase in number of publications towards this year. And you can also see that the highest popularity of these or skyrocketing these nanoparticles, lipid nanoparticles, became during the pandemic of COVID-19. We all still remember as we were at home, tucked, and then we get the vaccines, which actually were based on lipid nanoparticles, and they were delivering RNA to our body. So as I already mentioned, these particles are very versatile and modular. So they are now currently used and investigated in different vaccines, but also in gene therapies, because the advantage of this particle is that their cargo can be modified and also their surface can be modified to be more specific. So you can actually design these lipid nanoparticles to be able to deliver the cargo to the specific place within a human body. However, as these particles are also very versatile and modular, there are still some remaining key questions that we need to answer within LNP research. And before we move our research more for translational or for clinical trials, we need to first understand lipid nanoparticles on individual level. There are multiple different techniques that are actually allowing you to study these lipid nanoparticles at a bulk information. However, sometimes the information individual lipid nanoparticle might be much more useful compared to a full bulk results. So here are some questions that are still remaining, and they are important to understand when we are preparing lipid nanoparticles as a cargo deliveries, as a drug deliveries, for example, or RNA deliveries. So one of the questions is, for example, are my LNPs cargo positive? When you are loading or using encapsulation of your particles or lipid nanoparticles with RNA, you mix, and during your process, you actually don't know how many of your particles are RNA positive. And if you don't do any cleanup process, by measuring the RNA content within the particles, the free RNA in your solution will contribute to that information. So one of the questions is like, how many also LNPs actually contain the cargo? Because you might have a lot of cargo or a lot of RNA within one LNP, but it doesn't mean that majority of your LNPs will actually contain cargo. And for efficient delivery, one or the other might be more useful. The second thing, and it's one of the biggest problems nowadays in the lipid nanoparticle field, is their stability. How stable are these lipid nanoparticles? So if you go back a little bit, like 3 years ago in the COVID pandemics, if you remember the vaccines which need to be defrosted, they were delivered at dry ice, so very cold environment. Then they were defrosted, and they need to use in the same day. Otherwise, they were just discarded. So that tells you that there's still a big questions about the stability, how stable this particle can be, and also that's directly linking to storage conditions. There are multiple different storage conditions of these lipid nanoparticles, but which one is the right? Most of the lipid nanoparticles are stored with some sucrose, which could be 8, 10, 15% of your solution. However, the question whether that's a right storage condition is still unknown. And if you are having lipid nanoparticles which contain cargo in it, and you are measuring the concentration of cargo over time to study your storage or stability, you might still get the same information about level, but maybe your RNA or any other cargo is actually outside of this lipid nanoparticle. And then the last question is, when we are targeting specific receptors on the cell surfaces, for example, and this is in a context where lipid nanoparticles are actually also with a ligand loaded. So in this case, as I mentioned earlier, you are modifying the surface of these lipid nanoparticles, and by adding specific molecules, such as antibodies, which can then recognize specific receptors inside the cell. But in this case, you also would like to know how many ligands I'm having actually on the surface of each particle, or do all my particles actually have the same amount of ligand or not? So to answer all these questions, I will try to show you that by using single molecular localization microscopy, you are able to answer majority of these questions when you start to understand your lipid nanoparticles in a basic research level. But before we go there, I would like to give you some insight about single molecular localization microscopy and why single molecular localization microscopy when you study lipid nanoparticles. So to go there, we need first to understand where the lipid nanoparticles actually fit in a biological scale. So we all know that the lipid nanoparticles are very small, as it already says in the name, it's called nanoparticles. So you can see that the size varies, and it can be somewhere between 30 nanometer up to 150 nanometer. So as you can see here in this diagram, they actually belong to a size where you will find like extracellular vesicles or viruses. And then we now need to understand that if the lipid particles belong to here, what type of microscopy techniques we can actually use to see them. Because I'm seeing is believing. So basically, we would like to see how individual lipid nanoparticles look like with a microscopic scale. So to study these lipid nanoparticles, we need to understand which microscopy techniques can be used for it. So if you look on this diagram, I'm actually highlighting here different microscopy techniques. And none of them are the conventional microscopy techniques or the microscopic techniques which are detecting about the diffraction limit. And these microscopy techniques include turf microscope, which is total internal reflection microscopy, or epiluminescence, or actually confocal microscopy. So these are the much more common microscopy techniques. But if you realize these microscopy techniques are actually not able to visualize individual lipid nanoparticles because their size is smaller, and the limit of these technologies is at the diffraction limit of this microscope. So what is the diffraction limit? So diffraction limit is a size or wavelength where actually, or it's a size where you can still resolve two objects separate from each other. So in this case, if you use a conventional microscopy, it's roughly speaking half of the wavelengths what you are using. So it's currently is approximately 200 nanometers. And as you remember, I mentioned you that these particles are actually below 200 nanometers and usually are around 60, 100. So what other technologies we could use to study this particle? So one of the common technologies to study the size and the morphology of lipid nanoparticles is electron microscopy. And yes, electron microscopy, as you can see here on the bottom range, is a technology which has the highest resolution. You can detect particles, you can easily detect your lipid nanoparticles. However, the sample preparation, electron microscopy, might not be as straightforward. The other disadvantage of technology is that it's not a fluorescent technology, so it's very hard to get the information about your cargo or your cargo positivity or information about your ligand unless you are using some gold beads that can be detected by using the electron microscopy. However, there is this range of super resolution microscopy techniques. So these technologies are actually able to break the diffraction limit. So they are able to image below this 200 nanometers, which means that they are nicely in the range of our LNPs, so what we will want to look for. And one of these advantages of these technologies is that they are based on fluorescence. So by using 2-3 color fluorescent labeling, you can actually detect your cargo, your ligand, and also your morphology of your LNPs by looking on the surface markers. So one of these technologies, or one of the group of these technologies, is single molecule localization microscopy. So now I will give you some insight what this single molecule localization microscopy is, and then I will show you how actually we can use it to study lipid nanoparticles. So what is single molecule localization microscopy? So as it already a word says, it's a single molecules. So we are able to detect individual fluorophores. So how does it actually work? So first, when you have an object which is below the diffraction limit, so below these 200 nanometers, you will have fluorophores which are all blurred together. So when you use a confocal microscopy, you use usually a very low laser power. So that laser power, if you have antibodies, fluorophores bound to antibodies, those actually will switch on. So they will become to on state, which you can see here, and they will all overlap. So that will show you that you cannot actually resolve that object which is beneath here, because each of those will have, let's say, a radius of 200 nanometers, which will be a diffraction value. However, if you actually increase the laser power, so you are able, in this case, to switch on only subsets of these molecules. So when you switch on the subsets of these molecules, you can see things that you will start to see things sort of like blinking. You will see that individual fluorophores are switched on. And if you repeat this process multiple times, we are talking about thousands of frames or 10,000s of frames, but in a very fast rate. Then you are able to actually localize individual fluorophores. So individual disease does localize, put them down next to each other, and then you can resolve your image. So this is a way how you can, using single molecule localization microscopy, to break the diffraction limit, so go below the 200 nanometer and still resolve your image with the food, such as here, the logo of our nanometer. So this is a group, as I mentioned, it's a group of technologies. So one of the technologies which we usually use and Nanometer is capable to use is a D-storm, which is a direct stochastic optical reconstruction microscopy. So as it already said, it's direct. It's direct means that we don't need an additional boosting of a fluorophore to be able to get a stochastic switching on and off of our fluorophore of interest. And that's why it's stochastic, because it's pretty much random. You don't know which fluorophore will come on and off in a time. But if you use the right combination of a laser, dye, and laser power, dye, and buffer, then you will be able to achieve multiple switching off of this fluorophore, and as previously I showed you, then resolve your image and go below diffraction limit. So this technology has some advantages, and that's exactly showing you that you can study lipid nanoparticles, because the X and Y or spatial resolution is around 20 nanometer, and our LNPs are within the range of like 30 to 100, 150 nanometers. And it also can perform a 3D imaging, which in a Z resolution is around 50 nanometers. So we can get the very nice information about the morphology or the structure of our LNPs, but using multiple colors, we can actually also get an information about the about the ligand positivity, cargo positivity on these LNPs. One of the other advantages of the storm or this technology is that it also allows you a quantification of individual profiles. You will not get the perfect number of copies of your receptors or ligand on the surface of lipid nanoparticles. However, if you are comparing it between different conditions, such as if you are tuning the amount of PEG molecules on the surface of lipid nanoparticles, you are able to get an information whether adding more PEG molecules to your lipid nanoparticle composition is actually increasing the number of PEG molecules per lipid nanoparticles or not. So after this, having this small introduction to lipid nanoparticles and to a single molecule localization microscopy, I now would like to show you how you actually can do it. So what type of workflow you could actually follow here? So one key factor how stochastic optical microscopy works is in a TIRF mode. So TIRF is a total internal reflection microscopy, when actually you are using a very small evanescent weight of light within like 100 or 200 nanometer close to the cover slip. So this already tells you that actually you need to first capture your lipid nanoparticles to close the surface. So one of the 3 steps you need to follow prepare your lipid nanoparticles for a storm or single molecule localization microscope imaging is first to capture them, then you need to label them, and then you actually use detect them and analyze them. So as I already mentioned, first step is use a capture. So after capturing your lipid nanoparticles, now you are actually moved to the second step when you are performing some labeling. So what are the options what we can do here? So there are a few options. So first will be, we would like to label the surface marker. So labeling a surface of lipid nanoparticles will give you an information about their morphology and sizing. And then on top of it, you can actually also label your cargo detection. So mainly the cargo will be nucleic acids, which could be RNA or DNA. So you're using many of these dyes which are suitable for detecting nucleic acids can be also used. So you will have a second color. And then if your particles or lipid nanoparticles are actually functionalized and they contain a ligand on their surface, in this case, you can actually add an additional color, which will also give you a detection here. So in this case, you will be able to detect 3 different components of your lipid nanoparticles by one experiment in one single step. So what you can see here, you will get actually information about your cargo detection. You will get an information about your sizing, and you can also get an information about your ligand positivity, whether your particles contain a ligand and how much of it. And then the last step is actually a detection analysis. So in ONI, by using a nanoimager, we don't only provide a microscopy system, but we also provide an analyzing tool or acquisition tool and then analysis tool. And our analysis tool is actually a Cody platform. I will talk about it. It refers to collaborative discovery. So you will be able to even share your data with your colleagues without needing to use a small pen drive or get them access to your drive. So with that, I will go to show you today some examples where what you can actually do and how you can characterize the lipid nanoparticles by using single molecule localization microscopy. So as I mentioned earlier, you will get 2 informations. One will be a bulk information. So we get a similar information as from other techniques that which are currently used for sizing or also for cargo positivity. But you will also get an information about the single LNP level. Because at some point you would like to know information such as, oh, what is a correlate? Is there a correlation between the amount of cargo and the sizing? Or is there or when I modify my particles, when I use a different procedure to creating them, do I still get the same type of particle? Is the composition same or not? But today I will show you that with one technology, with one experiment, you can actually obtain a multiple different information. One of the information you can get is the sizing or morphology. So as you can see here, these are the examples. What you can see here in magenta, you can actually see an individual LNPs, which are imaged by single molecule localization microscopy. And please keep your eyes on the scale bar, you can see that these particles here are less than 60 nanometer, and these particles are around 70 or 80 nanometer. But in the same time, you can get an information about their cargo positivity. That's in the magenta. So in this case, we actually use the diffraction limited imaging, and then overlap of the diffraction limited image with a lipid nanoparticle will give you an information about your encapsulation or cargo positivity. At the same time, you can actually also look for a ligand detection, such as here at the top. You can see that there are some particles which contain your ligand. In this case is in the yellow, but they are also cargo positive. And nevertheless, the technology, and I will show you some examples of it, can actually be also used to test stability and storage and the formulation of your LNP. So in this case, I will give you an example where actually I made a thermal shock to the same LNPs and what happened with them. So what happened. But let's start with first with the sizing. So as already mentioned, the single colocalization microscopy is using stochastic blinking of the fluorophores, and that's why how we can actually break the diffraction limit. So here on the left side, if you look, you can see an example image, which is a small crop from our Cody system, so with our nanoimager, and you can see here that you have LNPs of different sizes. And by using machine learning algorithms, you can actually identify each individual lipid LNPs, and you can actually extract an information such as the size and have a histogram distribution of it. So you can see here, for example, on the top, this is a sample 1, when you can see that there are some like LNPs with this distribution around peaking around 80 nanometers. So the size of these lipid nanoparticles here in this subset is approximately 80 nanometers plus minus 25 as a standard deviation. And then, however, as I showed you, you can also get particles of smaller sizes. That's actually happening here when you can see that the smaller LNPs are actually around 45 nanometer plus minus 14, which tells you that these nanoparticles are much more uniform in their shape, which means that in the first one, you have a broad spectrum of them, but here you have it much more narrow. But what I really want you to point out is that if you will use a conventional microscopy techniques, such as confocal microscopy, you will not be able to distinguish difference between these two populations because the diffraction limit is here at 200 nanometers. So as I already showed you that this technology in one step can not only give you information about the sizing, but we can also give an information about the cargo positivity. And this is what I'm showing you here. So within the same sample, we label also the cargo, and you can see a nice overlap of a cargo signal, or in this case RNA, with your lipid nanoparticles. And when you look on a distribution of these particles, you can see I'm showing you two distributions here. The first distribution at top is a histogram distribution with the RNA particles or the intense or RNA abundance approximately around 1,000. So these are arbitrary units because this is fluorescent and it will depend on your intensity of laser signal. However, we optimize and recommend to use some specific laser powers when studying the lipid nanoparticles. So what you can see here, that this population here is a population which is RNA positive, and here below I'm showing you a population which is actually RNA negative. So this population doesn't have that information. However, if you would like to, most of the times, you would like to have a correlation between these two. So if you look on experiments or using other technologies, you can get an information about the size, and you'll get also an information about RNA positivity, but not all the time. All the time, you can actually make a correlation between these two. And what you can see here, that if you use these two different samples, one is a negative for RNA and one is positive, you can nicely separate these two populations from each other. And you can also see that there's a nice correlation between the LNP size and RNA abundance. So from this experiment, it shows that particles which are bigger in size are actually also containing RNA. And if you are using the same particles, then you might observe that some of the particles which are empty or they are RNA negative are actually smaller compared to RNA positive. But this is an ideal scenario when actually you have one particle which is bigger and already RNA positive, and then you have another particle which is smaller and RNA negative. But what about the scenario when you have sample when you don't know whether all particles are RNA positive or not. So this is an example here. So you can see here that there's an example of the same type of crop. And you can see that there are some particles which are RNA positive. So it's an overlap of the magenta signal with the cyan. And then you have also some particles outside which are smaller. But actually, they are very small. They're below 50 nanometer. So here you can see that within the same sample, our technology allows you to actually get information about the RNA abundance, but whether it's size dependent. So what you can see now here in this scatter plot, including the histogram of sizes and histograms of RNA abundance, is that particles which are RNA positive are actually bigger. The size of those particles is approximately, the median value is around 65 nanometer, but there's a broad spectrum of it because the standard deviation is 84. And you can already see it from the images. If you look here, all particles which have a cyan signal are mainly big. And then you have the other subset of particles, which is RNA negative. And those RNA negative particles are around the size of 37 nanometer, also with a broad standard deviation. But from our experience and imaging some micelles, we also observed that the size of the micelle is somewhere around 31 to 40 nanometer. So we assume that from this experiment, you can see that the particles which are nicely formed and contain RNA are real LNPs, and the smaller particles are actually particles which are RNA negative and maybe correspond to micelle peak. Whether they are micelles or smaller RNAs, it will be smaller LNPs. It actually depends on your sample. So this is a scenario when I'm showing you that the technology within the same sample, you can, by using the single localization microscopy, distinguish two different subsets, one which is RNA positive and one which is RNA negative. But what about the encapsulation or positivity? When we have a distribution such as here of the particles, we still would like to know what's the percentage of particles that are RNA positive and what is the distribution of those. So you can see here that the positivity of these particles is almost 50-50 by looking on the image, but we would like to have some quantitative information. And this is what I'm showing you here by using two different samples. In the first sample, you have particles which are RNA positive, but they're also particles which are negative. And in this case, by using our technology and imaging multiple different fields of view, you can actually see that the positivity here was only 64%. Each dot in this graph represents a different field of view. And usually per field of view, we are getting approximately 2,000 LNPs. So by using 10,000 LNP, using 5 fields of view, you can achieve approximately 10,000 LNPs. And in this case, the 10,000 LNP already corresponds to a bulk experiment and give you a good statistic. On the middle graph or middle example, you can see that these are LNPs which are bigger and they're all RNA positive. And in this case, in this sample, was highly positive for RNA, and the percentage of positivity was around 92%. Which means that if you're looking, if your question is, do all my particles contain RNA? The answer here will be, yeah, your majority is actually having RNA, but in the scenario of a sample 1, that's not true. So in the scenario of sample 1, the RNA positivity is only 60%. So some of your particles are RNA negative. So if you take these two samples and both will have the same RNA, most probably the 92% will have a higher efficiency to move to clinical trials, or like with a high probability to move to clinical trials compared to one with 64%. It also tells you that your sample preparation in the case of particles with 92% positive is much more accurate compared to one of 64%. But also, I pointed out that you can use this technology to study stability and storage conditions. So here I would like to show you an example when I use the same LNPs, which size was approximately around 98 nanometers. So this is please have a look first on the green histogram. So these are the subset of LNPs which actually are very clean, they're all positive, and you can see that the size is approximately 98 nanometer with a standard deviation of 31 nanometers. And then what I did, I performed a thermal shock. So I actually simply took the vial of these LNPs, I boiled them up, I boiled it for like 5 minutes, and then I performed the same assay. And in this case, what I observed, that these particles slightly become small, they were completely disrupted. I lost the RNA signal, so they became all RNA negative, and the size became small, somewhere around the range of 38 nanometers. Here in the images, you can see the positivity of this particle. So from 86% positivity, so 86% of LNPs containing cargo, I dropped to 5% only, which tells you that by using this thermal shock and using the technology, you get an information here about the bulk information of your LNPs, but at the same time, you get information individual LNP level. And our technology and our software also allows you to have a look on individual particles. So if you use our ONI system and our Cody platform and analysis, you'll be able to click on individual LNPs and get an information about their abundance, about their size, and also whether it contains a ligand or not if you use the 3-color imaging. So by showing you that you can use actually this technology to study in one experiment, to study sizing, cargo positivity, also storage conditions, I also would like to show you that you can also look for a ligand specificity or a ligand abundance. So this is a scenario when you modify your LNPs to be targeting, to be prepared to target specific target. Most of the time, these LNPs are modified by using antibodies, which then recognize some receptors on other cell surface, and by these they can get internalized to the cell and the RNA content released. So here I'm giving you an example where you can see a 3-color imaging when we label the surface marker of LNPs, we label the ligand, and we also label the cargo. And what you can see here, only just from images, the particles, there are some particles which are triple positive, which means they contain cargo, ligand, and surface. And there are some particles which are actually cargo negative, but they contain basically ligand and surface marker. The big advantage of using ONI's Cody platform is that we are performing 3-color colocalization, or we call it like positivity. So you get an information, what's the population, what's the percentage of population in my sample that is only have a surface marker, which one has just a ligand, or which has a cargo, but you also get all the other combinations. So for example, here on the right side, you can see that if you use a sample when you're having a label, in this case, you had labeled all 3 things, you can see that only 5% of your lipid nanoparticles are containing the cargo. So only 5% of those have actually cargo. But then around 20% of those there are particles which contain ligand. But what's more important is that you have 40% of particles which contain cargo and ligand, which means that if you want to answer how many of my particles are actually ligand positive, and in this case is a 20 plus 40, so 60% of your particles are ligand positive. But only 45% of your particles are cargo positive. So you have some particles which is distinguished here, it's like 15% difference, which are particles which only contain ligand. So by this, I would like to, after showing you all this, I would like to also move and give you a little bit insight to our data analysis of these lipid nanoparticles, because seeing is believing, but we also would like to have some quantitative information of it. So how we at ONI perform quantitative information. So to do that, after acquiring your data, you can upload your data on our Cody. So Cody is a collaborative discovery platform. That stands for collaborative discovery. The interface looks like this, what you can see on the top. And after uploading the data, you can perform multiple steps to perform the analysis. And that's how you get the information. And if you open an individual data set, such as here, you can see that there are some different steps. So briefly, in the first step, you will do just a drift correction. This I mentioned that this technology requires imaging multiple frames. So when you image for a long time, let's say 5 minutes, you start to see a drift. There's a thermal drift, so your sample starts to move, so we need to correct for it. So software does it for you. With the second step, you can perform some filtering parameters. So sometimes not everything is a single molecule. You can have some small background. So this is the point when you can set this up. The third step is actually identify what is a cluster. So we have a cluster analysis algorithm, which are based on machine learning, but also by DB scan. you can using a surface marker to determine what is an individual LNP. And after determining what's an individual LNP and measuring the size, we go to the quantitative part of it. And this is this type of widget what you can see here. When here on the bottom, for example, will show you what percentage of these particles are actually pan-LNP, or in this case, it's like a surface marker, or which are cargo and ligand positive. And at the end, you can actually download all this information. And when you download this information, you end up with a CSV or a text file, which you can open in Excel, and you can plot, you can get a multiple different information. You will get a centroid of each cluster. You will get whether the cluster is cargo positive or ligand positive, and you will also get a lot of information about the abundance, number of localizations, and you can perform different filtering and create the graphs which I just showed you earlier more about. So by this, I would like to summarize. So today I showed you why actually the single localization microscopy could be used to study your lipid nanoparticles at the basic research level. So by using one experiment, one preparation, one acquisition, and one data analysis, you can get an information about your morphology and size of your LNPs. You can get information about your encapsulation or a cargo positivity to give you an information how many of my LNPs are cargo positive and whether there is some correlation between size and cargo positivity. Then you will get an information about stability and storage condition. So if you are interested whether your sample is stable at 10% sucrose or 15% sucrose, you can perform this experiment. And if you start to see that your LNPs are falling apart, you start to see a smaller peak around 30 nanometer, which could correspond to the micelle size, and they will be RNA negative. But also, if your LNPs are more ligand specific, or you are targeting a specific receptors on the cells, in this case, you can perform a 3-color imaging, and you'll get an additional information about the ligand detection, about the number of molecules or number of localizations of the ligand per LNP, and whether the different ways of preparation of LNP actually increase the amount of ligand on their surface, or actually, in contrary, it decreases. So with that, I would like to thank you for your attention, and I'm ready to answer some questions.
Great. Thank you so much for your presentation, Stefan. So we're now going to open the floor for questions for Stefan, which he is going to be answering live. So once again, if you do have any questions, please feel free to just drop them in the chat box. Looks like we already have some questions submitted. And our first question is, what ligands can you detect, and do they have to be human?
No, so in terms of ligand detection, it can be customized. So the system allows you to do 3-color imaging, as I showed you. So usually for a cargo positivity, we keep one channel. For a surface marker will be a next channel. And you can use a third channel for your ligand detection. So there are 2 ways. One, you can pre-label your ligand with a fluorophore before your formulation. So before you make them incorporate to your lipid nanoparticles. That's one option. Second option is that if your ligand is an antibody, then if your antibody is, for example, a human or mouse or rat or rabbit, you can just use like a secondary probe to detect it. So for example, you can use a secondary probe conjugated to fluorophore, such as a Fab2 fragment or full secondary antibody. And yeah, basically you have these 2 options. So we are not really limited. You're not limited if you're looking for a ligand detection. It's just the right way to use the right target for it. So if it's an antibody, you can always go with the secondary probe against it, or you can directly label your ligand or your antibody before you make them in the formulation of the LNP.
Great. Kind of related to the topic as well, can you just expand a little bit? What do I need in order to detect my ligand?
Well, so first of all, actually, you need your LNPs, which contain the ligand. The second thing is that it can be always customized. So if your ligand is an antibody, you can actually conjugate your antibody directly with the fluorophore. and then make the combination, make your encapsulation or make your integration of the ligand to your lipid nanoparticle. So that's what you can do. The second option is you just perform the same labeling as you would do for any confocal imaging for any receptor. So for example, if your ligand is an antibody, that's a human antibody, then you can use a secondary Fab2 fragment against the human, which will be labeled with the fluorophore. So in this case, in this example, which I showed you, we use a 560 channel for it to detect the ligand. So basically, there are a lot of options which we can explore, but it will at some point could be custom made, or we can customize it depending on your ligand and your targets.
Okay. Looks like this question is a two-part question, Stefan. Someone said, hi, I don't know if I missed this point. The Cody analyzer is available also for images acquired without ONI technologies. And then another question is, do you have with the ONI system good SMLM also for fluorophore 488?
Yeah, so start the first question. Unfortunately, the answer is no. So the Cody platform is fully integrated with the data set recorded by Nanometer, so our own system. So unfortunately, cannot support other files at this stage. And the second question, in this experiment, we prefer, as it's not easy to get the fluorophore for the 488 channel, or which performs very well with a single molecule precise microscopy, we are using the 488 channel to detect the cargo. And that's actually a diffraction limited, as you can see here in the cyan. So in this case, we are using the 488 channel as a diffraction limited channel.
Excellent. Someone asked, can you detect a cargo other than RNA?
Yes. I mean, if we look for nucleic acids, in this case, you can detect RNA, DNA. You can detect anything. If you have a peptide, if you have antibody or whatever it's it, unless you're able to label it. So the key factor here is that you need to be able to label it. So in this case, if you use a nucleic acids, you can have a dyes which are specifically binding to nucleic acids and then can be specific for different laser lines. But in the case of, let's say you have a peptide or your cargo, it can be your cargo, can be even an antibody. In this case, if you pre-label your cargo and before you encapsulate it, then you can detect it. So I will need to know more information whether what is your cargo in this case, but most of the time it can be, if it's pre-labeled, we can detect it.
Excellent. Someone asked a very general question. They said, how long does the whole process take?
Oh, so our stuff is that if we use a nanometer, the field of view allows you to get approximately 2000 LNPs. So in this case, that will take you more or less 3 to 4 minutes. So you can assume that if you want to make a statistics from 10,000, so it's 5 times 3, around 15 to 17 minutes, and you get an information from approximately 10,000 LNPs.
Excellent. Okay, next question. What format do my LNPs need to be in for this workflow?
Well, it's a good question, because most of the times, as different companies or different labs are having a different LNP composition, and also the storage conditions are different. So some people use a tris buffers, some people use a PBS, some people use a sucrose and different concentrations. So in this case, you don't need to have your LNPs need to be prepared in the format as you are having. Most of the time, the concentration or the dilution factor, what we are using here, will depend on the concentration of your LNPs. Or sometimes also we can guess based on the concentration of RNA, but it will be a dilution factor will be somewhere from 1 to 2000 up to 1 to 50,000. So there is no specific formulation or specific storage conditions that you need for attaching your LNPs and study them by single molecule quantization microscopy.
Great, thank you for that, Stefan. The next question is, how many LNPs do I need for this assay?
Yeah, so this is a tricky question, but I can tell you that if approximately if your RNA concentration is somewhere around 0.1 mg per ml, so 100 micron per ml, you will need to do a dilution 1 to 50,000. So you can see that the dilution of your LNPs is very high. So you will need like 1 microliter to 50 ml of a PBS in this case, or it can be your storage buffer. So you don't need too many particles. And also you don't need too many particles because if you are using our ONI chips, we have a 4-lane chips and the volume of each of the lane is approximately 5 microliter. So you really don't need a lot of LNPs to perform this assay.
Okay, the next question is any fluorescent dye compatible with this assay?
The answer is no. So unfortunately, single molecule localization microscopy, and one of the drawbacks of it is that they require imaging buffers, so different specific conditions that you need to use to make molecules switch on and off by using high laser powers. So make the molecule switch on and off, you need to have buffers which contains thiols and glucose oxidase. And therefore, there is a limited number of fluorophores. However, in ONI, we have a list of fluorophores which are compatible with our nanoimager and with our imaging buffer. So if you go to our webpage, you'll find a list of fluorophores that are compatible and with our imaging buffer. So that's how it is.
Excellent. All right, and the next question is, can you track LNP uptake by cells?
Yes, the answer is yes. So if you are pre-labeling your LNPs, our system is not only ready to study lipid nanoparticles, but it's a versatile microscope system which can work in a turf mode with the low laser powers. And also the chamber can be heated up to 41 degrees. So you can create real physiological conditions by warming your chamber to 37.5 degrees. In this case, you can just place your cells on a microscope, find your focal plane, so find your cells, put your LNPs, and gently image in a time-lapse image. You can do also different fields of view and look for an uptake of LNPs by using cells. Yeah, it's possible.
Excellent. Well, thank you, Stefan. It looks like that was all of the questions that we had from the audience. So thank you again. If you do have any questions that Stefan did not answer during this time, you are more than welcome to email us and send those questions to us, and we will make sure to get them to Stefan to answer for you. But once again, this webinar was recorded. Therefore, if you need to refer back to it, you absolutely can. All these recordings can be found within the resources section of our website, so make sure to check that out. Thank you to all the attendees for joining us today. And upon exiting the webinar, you will receive a survey. Please fill out that survey just to provide us feedback on your experience today, or tell us if you're in need of being contacted directly by ONI for more information on our offerings. Thank you again, Stefan, and thank you again to everyone. And we hope that everyone has a great rest of your day.
Thank you. Bye, everybody.